For ReachMD, this is Audio Abstracts. The following is produced in collaboration with the Crohn's and Colitis Foundation. I am Dr. Damian, Maseda, an immunologist and research associate at the University of Pennsylvania, and a member of the Crohn's and Colitis Foundation, Rising Educators, Academics, and Clinicians Helping IBD Group, or ReachIBD. Today I'll be reviewing an article recently published in the Inflammatory Bowel Disease Journal titled Iron Sequestration in Microbiota Biofilms as a Novel Strategy for Treating Inflammatory Bowel Disease. The precise role of the microbiota during IBD is currently a matter of intense research. Many studies have addressed whether the microbiota composition and diversity are altered in patients with Crohn's disease and ulcerative colitis compared to healthy patients. Some results, like the reduction in microbiota diversity during IBD, are highly consistent overall across institutions and studies. But there are also many important unresolved questions as to how the microbiota can contribute to inflammatory intestinal damage and what role the key micronutrient iron plays for pathogenic bacteria during colitis. That's why the authors of this study aim to understand two things. First, if the capacity to generate biofilms by human mucosa-associated microbiota was different in healthy and IBD patients, and second, whether a novel derivative of the anti-inflammatory frontline therapy mesalamine would alter these biofilms and their virulence in vivo. In order to evaluate differences in iron intake from the microbiota, the authors established a model for in vitro biofilm generation from stool samples and quantified intracellular iron concentration. They then combined these biofilms from either healthy or IBD patients with human intestinal epithelial cells grown in vitro to address the impact of the different biofilms in causing damage and to promote the expression of inflammatory markers. They also used 16x sequencing to evaluate the impact of two novel iron-chelating derivatives of mesalamine called ATB428 and ATB429 in the composition of the microbiota. Additionally, a mouse model of colitis was used to evaluate the toxicity of microbiota-derived biofilms and the properties of iron-chelating derivatives of mesalamine in protecting against colitis development. The authors of this study first found that the microbiota from IBD patients can generate larger anaerobic biofilms and also exhibit larger iron intake than the biofilms from healthy individuals. The authors then evaluated if these altered biofilms would have differences in their virulence. For that purpose, they co-cultured biofilms with human intestinal epithelial cells and found that bacteria from IBD-derived biofilms were able to translocate in larger numbers and with more invasive properties than bacteria from healthy individuals. Interestingly, bacteria from Crohn's disease and ulcerative colitis patients produced fairly similar results. The authors then found that both of the novel hydrogen sulfide-releasing mesalamine derivatives chosen for this study were able to effectively chelate iron. This property allowed ATB429 to reduce the intracellular iron content in bacteria from biofilms. Surprisingly, this drug did not have any effect in the microbiota or biofilm composition. However, it did significantly alter the purine and pyrimidine metabolites used by the microbiota, which are dependent on iron intake and are important for the colonization of host tissues. Furthermore, iron concentrations in fecal bacteria from mice treated with DMBS to induce colitis demonstrated increased concentrations of iron. Finally, administrations of ATB429 protected DMBS-treated mice by diminishing bacterial translocation to the liver, intestinal pathology, and invasion of the intestinal mucus layer with concomitant addition to the epithelium. In addition to these findings, this article also suggests that there might be a link between increased iron requirements in the microbiota of IBD patients and anemia in their host, although more causal evidence is needed. Further studies focus on how certain drugs can alter iron chelation and metabolism in the microbiota would be needed to corroborate these findings and expand our knowledge of the interaction of the microbiota metabolome and its role during IBD. In summary, the current study demonstrates that microbiota biofilms grown from IBD patients have an increased capacity to assimilate iron that correlates with increased pathogenic features in intestinal epithelial cells. The study also provides preliminary evidence for a novel hydrogen sulfide-releasing mesalamine derivative to exert iron-chelating properties and reduce gut microbiota biofilm virulence. This translational and integrative study opens interesting aspects of therapeutic manipulation through a deeper understanding of the interactivity between microbiota and anti-inflammatory drugs in current use for the treatment of IBD. Thanks for listening. If you're interested in this topic or others on Crohn's disease or ulcerative colitis, the Inflammatory Bowel Diseases Journal, which is the official journal of the Crohn's and Colitis Foundation and supports the Foundation's missions, brings the most impactful and cutting-edge clinical topics and research findings related to inflammatory bowel diseases to clinicians and researchers. For more information on the Foundation, please visit crohnscolitisfoundations.org. This has been a presentation of audio abstracts produced in collaboration with the Crohn's and Colitis Foundation. For more information and for reference links of this article, visit reachmd.com/foundation.